Okay, so we're going to start by talking about karyotypes. And to see what makes us different from other animals and to see what makes us unique, we have to look at our own human genome. So first off, what is a genome? Your genome is your full set of genetic information, and it's that any organism is carrying. Obviously, this is located in its DNA. So when we're looking at a genome, we have to study the actual chromosomes. And to see these actual human chromosomes clearly, we can photograph the cells when they're in mitosis using a special camera, when the cells are fully condensed, and that way they're easy to see. Then you can have people that will cut up those pictures of the chromosomes. They cut them out from the pictures, and then they arrange them into something called a karyotype. A karyotype is going to be the complete diploid set of chromosomes, and that's going to be when they're grouped together in pairs, and then they'll arrange them so that they decrease in size. So let me show you what that looks like here. So here's an image of a karyotype in a typical human cell, right? So we see that there are, hold on, where's my pointer? Total of 46 chromosomes, right? There are 23 pairs. We can see, yep. Here's one pair, two pair, all the way up to 22 pairs over here. And then we have two more, 1, 2. So total, if you were to count all of these up, you would count 46. We see that there are, the total of 46. So keep in mind that the sperm, they are a haploid cell, meaning that they're going to have 23 chromosomes. And then our eggs will be the same. So that means that when we combine our sperm and our egg, we're going to get a diploid egg that now when it's combined with the sperm with two sets of 23 chromosomes for a total of 46 chromosomes. And again, keep in mind that 2 of those 46 are going to be sex chromosomes, which will determine the individual sex. Right? We know that females, they have 2 X chromosomes, versus males, they are going to have an X chromosome and a Y chromosome. And the reason this is the case is because all eggs, they're going to always carry a Y chromosome. And sperm, about half of them are going to carry a Y, and then half of them will carry an X instead of a Y. So half of the zygotes are going to be female and half of them will be male, and that's kind of why we have an equal number of males and females on our planet. Just really quickly, differences between X and Y chromosomes. An X chromosome will have about 1,200 genes on it, and the Y chromosome is much smaller. In a Y chromosome, we only have about 140 genes. Most of those genes are going to be related to male sex determination and also will be related to sperm production. But getting back to overall picture here, all of the chromosomes beside the X and the Y, so the other 22 pairs, all of those other chromosomes are what we call autosomal chromosomes. Those are the remaining 44 chromosomes. So when we're talking about an individual, we're going to talk about them, if it's a human, as having 46 chromosomes. And then after it, we'll say that they are either XX, or we can say that they are XY. And so an individual that's XX, that'll be a female, versus this individual will be a male. There are instances where you can have chromosomal disorders when there are errors in meiosis. And I'll talk about that in our next screencast, but here I want to stick to talking about karyotypes and something called amniocentesis. So what is amniocentesis? Well, it's a pretty cool technology that many of your parents went through. You probably just didn't realize it. You would not have a reason to know what it is because you're young and you haven't had children. But the whole point of amniocentesis is so that you can create the karyotype, right? So if you look back on this slide here, this whole thing, remember, is a karyotype. Then once a karyotype is created, you can have a technician look at it and tell whether or not there are problems. And what kind of problems are we talking about? We're talking about problems that occur within a developing fetus. So amniocentesis, this is a process that's done on pregnant women. Let me show you here sort of what that process looks like. So a little graphic here, but let me explain to you what is going on. So we have a developing fetus, and then we also have this thing here, an ultrasound, that allows us to carefully see what's going on inside, and the doctor will perform this technique to help us see what's going on. And then we have this massive needle here. And that has to, it doesn't show it particularly well, but if you think of your stomach right between here, you have all of your abdominal muscles. So that needle has to go through, has to penetrate through the abdominal wall of the woman, but then it also has to go through the wall of the uterus, and then it has to go into the amniotic sac, which is shown here, kind of outlined in purple. And the amniotic sac is going to be what is surrounding the developing fetus. A fetus, when it's developing, is going to shed a bunch of its cells. And this is similar to the way that we, as we walk around, are shedding our skin cells, and they just kind of fall off our body. But when a fetus is shedding these cells, they're going to stay alive because they're in actual amniotic fluid, all of this stuff in the purple around the fetus. And that amniotic fluid contains a lot of nutrients, and it basically has perfect conditions for growth. The actual cells may not grow, but they definitely won't disappear because they're being fed. The cells, we can go into the amniotic fluid and find a lot of these live cells, and we know that then they are going to be genetically the same as the cells of the fetus. So they're body cells from the fetus. So using this needle here, we're going to extract about 20 milliliters of the amniotic fluid, which sounds like a lot, but it's only about 4 tablespoons. And in about 2 days, the fetus will be able, or the woman will be able to replace that amount of amniotic fluid that was lost. And then we have our puncture that came in through here in our abdomen and the actual fetus. Amniotic sac, and so those will seal up pretty quickly as well. So basically the doctor just has to use this ultrasound to make sure to not actually stick the needle into the fetus itself. And the ultrasound will help us to know where the fetus is so that we can make sure we're getting the fluid and not its brain juice. Once the amniotic fluid is out, we're going to put it into a test tube. But remember that this also contains a lot of cells. Okay, I'll draw those in green there for you. Those are the cells from the fetus. But we want to separate it out so that we only have the cells. So what we do is we use something called a tissue culture flask. Sorry, that's a very bad picture of it, but basically it's a flask that goes on its side. And then we're going to add the cells into the tissue culture flask to grow them along the bottom of the flask. And we're going to grow the cells until they reach confluence, which means that they're going to grow so close together that they're going to be touching. So that's a lot of cells. Then we don't need the rest of the liquid that we originally had because we have a bunch of the fetal cells. But before we actually extract the cells from the tissue culture flask, we're going to pour in a particular chemical. The chemical is called colchicine. And this is something that's extracted from the saffron plant, in case you're curious. And colchicine is a chemical fixative. And what fixatives do is it allows the cells to grow and go through interphase and then go through the S phase so that we have the duplication of chromosomes. It gets it all the way up to prophase of mitosis. And then the chromosomes get condensed so you can see them easily. Then we can stain it so you can see the chromosomes with the naked eye. And the fixative is basically locking it up so that there is the cells don't go through metaphase, anaphase, and telophase. And again, that means that the chromosomes are condensed and visible to someone that's looking at them through a microscope. Then the person that is doing this, we'll take a photo of the chromosomal spread. So if that's our cells with our clearly visible chromosomes inside, they'll take a snapshot, take a picture of that, and then what they'll do is they literally take scissors and will cut out the individual chromosome pictures. When they're done, it will look like what we saw previously with a karyotype. And remember that we're arranging this in a way so that we have the longest ones. We have our longest chromosomes up top, and then the shortest ones are down here at the bottom. Keep in mind also that we arrange them so that they're with their homologs. They don't have to be with their sister chromatids and be a full X at this point. It could be half. But keep in mind that, we have one here is going to be from mom, and the other one will be from dad. And you should always have pairs within these 22 chromosomes. And we can see that as we go through, each of them does have a second pair to it for chromosomes 1 through 22, except for the sex chromosomes, which are not going to be paired up. So here we see that we have two X's, so that would mean XX, that is a female. And if you were an individual that got this karyotype back, this would be a good thing. We don't see any chromosomal abnormalities or any syndromes. Let's look quickly at a male karyotype just to look at the differences. We see here, obviously, we have no Y chromosomes and two X's. Now a male's karyotype, we can see that there's one X and one Y, so that makes it a male. And again, as we go through, we see 2, 2, 2, all the way through we have pairs that match up, so this is another healthy baby boy. But life isn't always perfect, and sometimes you have these syndromes that can occur if you have issues with chromosomal abnormalities. So we could have something called aneuploidy, which again I'll go into in the next screen class, but that basically just means that we have an improper number of chromosomes. Okay, so if we go through, we see, okay, we're good, all the way through, doop, 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 all of those have 2, but when we get to chromosome number 21, we see that it has, in fact, 3 chromosomes instead of 2. And so this issue with chromosome 21 having 3 chromosomes, this is an issue where the egg had something called non-disjunction occur. Again, more on that in the next screencast. So basically 2 chromosome 21s from mom entered into fertilization, and then the sperm added 1 chromosome 21, creating 3 of them. And so this is what we call trisomy 21. And some of you know that this is also called Down syndrome. The word tri means 3, so 3 chromosomes. When you have issues where you have extra chromosomes, and when there are 3, you call it a trisomy. If we had 3 of chromosome 20, we would call it trisomy 20. We'll talk more about another trisomy in just a minute. But again, we probably all know someone with Down syndrome, or you've seen someone with it. And people with it can be perfectly happy people, and they can be productive members of our society. And while we may think of it as something that's pretty severe, it's actually one of the least severe chromosomal syndromes that you can get. And so when a parent gets this karyotype back, they have to make a decision. Some parents might decide that they want to terminate the pregnancy, and by that I mean have an abortion. Others may say, I'm perfectly fine bringing an individual like this into the world, and that's going to be a decision that you guys will have to make if you decide to be a parent later in life. So let's look at something else that is worse than trisomy 21. So this individual, looking at their chromosomes, we see that they have 2 X's and no Y, so this is going to be a female. But here the main issue is going to be on chromosome 18. And if we have 3 chromosomes instead of 2, we call that what? That's a trisomy. So this is trisomy 18. Also sometimes called Edwards syndrome. Again, here in chromosome 18 we have a trisomy, so 3 of chromosome 18. So there are some really graphic images that you can find of chromosomal abnormalities, but here I'm going to show you some that are a little bit more mild If you want to go find something that's a little more graphic, feel free to Google yourself. So as you can see, this child has some severe formation issues, the legs being twisted, and usually individuals with Edwards syndrome have severe mental retardation. Usually they won't live more than like 3 months. Some are miracles and can live several years, but again, it is such a huge debilitating thing. You can often also have major structural issues to the brain and the skull, which can, of course, cause death. Another syndrome that I want to mention is something called Patau syndrome. And in this instance, the egg, again, has messed up and has led to that thing called non-disjunction. And we, in here, will have a trisomy 13. We can see that, again, on 13 is where we have the triple chromosome, creating that trisomy. And Patau syndrome is more severe than Edwards. So I'll show you a picture here of a child's head and face, but just keep in mind that if this is how incredibly affected the face and head are, imagine what sort of the rest of the body looks like. Again, this is a very mild picture of Patau's. There's some really sort of scary stuff that you can go and search for on your own if you'd like. Most people who have Patau or Edwards syndrome, typically the pregnancy will terminate itself because there are so many issues. But if it does not, then the child will live for a maximum of, a few months to maybe a year or so. But in most cases, having Patau's or Edwards is basically a death sentence. So a theme that we talked about here is basically how there's this problem with the egg cells. So why are we blaming the egg cells? A woman is born with all of her eggs, and they're basically in suspended animation. So they haven't fully undergone the meiosis events that are needed to develop into the egg cell. But, however, they've gone a pretty good way towards that direction. But keep in mind, if an individual has eggs sitting around in suspended animation for a really long time before it's released into the fallopian tube and is fertilized, right, it may be close to 25 to 30 years. And that's a lot of aging, right? If you think of, have you owned anything for 25 to 30 years? No, because you're not even that old yet. But if you do, having something around for that long, it won't really do so well. So that's why amniocentesis is something that's often done for people who are 35 and older. And this is because the egg cells are often will have trouble going through meiosis too, and a lot of mistakes can be made. So the spindle fibers may not do as good of a job connecting to those kinetochores, and they may not pull the chromosomes apart correctly. So when an individual is 35 plus, it's suggested that they do an amniocentesis so they can be prepared for what you might do if you find out that you have a chromosomal abnormality in your fetus. And the last thing that I want to talk about is these disorders we've mentioned previously have been on the autosomes, right? The chromosomes 1 through 22, and not the sex chromosomes. But you can have non-disjunction and problems in the sex chromosomes. And we can have this non-disjunction in the sex chromosomes create it so that instead of having either an X and a Y or an XX, instead you could potentially have 3 X's, or you could just have 1 X. You could have maybe 1 X and 2 Y's, or 2 X's and a Y, right? So all of these situations that I have listed on the right makes a big mess of things, right? So trisomy 21 or Down syndrome, we know that tends to have some pretty mild, pretty mild of the syndromes that I talked about. But the sex chromosome abnormalities are probably less debilitating. You probably have peers, even here at Choate, that may have some of these chromosomal abnormalities of their sex chromosomes. So they may not know it, and you don't know it. It's pretty hard to notice. But in extreme cases, perhaps. And the individuals typically don't know that they have this chromosomal issue until they try to have children themselves, and then they realize that they're having trouble because they have too few or too many sex chromosomes. So I can tell you more about that stuff if you're interested in what that actually looks like. It's pretty cool, interesting stuff. But that's it for now.